Hi, my name is Sam, and today I'm going to present to you our work on assessing the hemodynamic transfer function at a frame rate of 5 frames per second. And as you can see in the title, we use this trajectory we call T-Hex Spiral-In trajectory. So this talk, which basically means that this talk has two aspects. It has an aspect of the technology we use to acquire our data, and it has the aspect of showing you how this can be useful to measure the hemodynamic transfer function. I have to mention that I'm showing quite early results in this project, but I think there are one or two interesting things which I can already show you. So I would like to start by showing you why we think it is interesting to assess the HRF at a high sampling rate. So here I show a picture which I adapted from a paper by Polimeni and colleagues, where they presented an oscillating stimulus to the participant in the scanner. And then they measured the magnitude of the response to this oscillation in the brain. And what you can see at high sampling rate or at low frequencies, this magnitude is well predicted by the canonical HRF. However, if we go to higher frequencies, we see that there's basically a factor of roughly 80 missing at 0.75 Hertz. And what you can then directly calculate to recover such an effect, you would need a TR of 0.67 seconds at least to recover such an effect according to Nyquist sampling theorem. Another paper that we found interesting was this work by Dowdle and colleagues, where they measured the difference between the HRF during a fixation and a phase detection task in the primary visual cortex. And what they show is basically on a TR of 1 second, you have here around the violet areas barely a difference between the different HRFs. However, if you lower the TR, you immediately see that there was a difference between the two HRFs, which could only be recovered at a fast sampling rate. So based on these two references, I could hopefully already convince you that the measurement of the HRF profits from a fast sampling rate. So here is this trajectory, this T-Hex Spiral-In trajectory, which we used. You can see that it is a spiral-in trajectory just from the fact that it sort of directly moves outwards in k-space and then slowly circles inwards. It is 3D trajectory, which means it covers multiple planes of a 3D k-space in one shot. And we just decided to use the spiral readout because it gives us this extremely efficient readout. So it transfers k-space at a high speed, which we need to reach the 0.2 seconds per volume, which we have here. The trajectory pierces through this T-hex or tilted hexagonal grid I'm showing here. So the shot I've shown before lies in this area marked here in red. And the good thing about this grid is that sampling points on this grid are spaced uniformly, which leads to a uniform but wide spacing of aliases in image domain, which then in turn leads to a well-posed inverse problem we need to solve during the image reconstruction. You can already see that our field of view is large enough to cover the whole brain. The image reconstruction we used is also a little bit a deviation from what we usually do. So here I'm showing what you need for classic sense reconstruction. You need a coil sensitivity map and your coil signal, and you can basically just write the reconstruction as an inversion of a linear system of equations. To make our method work, we need a little bit of an extended signal model. So what we also have in there is an off-resonance map. Spirals are just sensitive to off-resonance effect. That's why we need to account for this in the signal model. And we have this field camera we placed in the scanner, which is equipped with 16 MMR probes and just measure the gradient fields that are effective in the scanner. And we can also use this measurement and plug this into our signal model. And if we discretize the signal model, we're again just solving a system of linear equations during the image reconstruction. Here, just as an example, such that you get a feeling that this produces good images, I'm showing the mean image of all 3,000 images we acquired here in 10 minutes. So you can see we have gray-white matter contrast in the images. We see structures like the ventricles. And really the field of view is quite large. So here we cover the whole cerebellum, but also like the whole cortex is covered by this field of view. We used an event-related design. So we presented happy faces to the participant with happy and sad smileys on the left and on the right. And because here the smiley on the left is happy, the participant had to press a button with its left hand. I'm showing here only some single subject data, which is just the start in this project. We analyzed the data in two ways. First of all, we started out with a very classical GLM approach, where we used the informed basis set to build the regressors which are related to the task. And with that, we could basically get T-score maps to investigate where do we have significant activation in the brain. Based on that, we defined 6 regions of interest. I'm going to talk about that in the next slide, where we could already measure the AHRF, which we found based on this informed basis set in the GLM. We then also went on and replaced the informed basis set we had in the GLM with an FIR basis set, which was extremely flexible thanks to the high sampling rate. So we could use 160 bins in this FIR basis set. And with this, we obtained the second measure of the HRF in these regions of interest. And we then wanted to compare how these fits look and what can be accounted by the informed basis set and what can only be accounted by the FIR basis set, and whether we see benefits of the high sampling rate in the fit. So first, I would like to show you the T-score maps we obtained for activation related to the button presses. So we found activation down here in the cerebellum and also here in the left and right primary motor cortex and somatosensory cortex. This is nothing unusual. This is basically just what we expected as activation patterns for button presses. So here I can now show you the results of the HRF analysis. So on the right, I have the fit of the informed basis set, and on the left, I have the fit of the informed basis set, and on the right, I have the fit of the FIR basis set. And in the center, you get this sort of graphical legends where I just show where the regions are, which I'm talking about. So here in the left primary motor cortex, you already see that the FIR basis set shows a steeper slope or decrease from the peak than the informed basis set. And steep slopes are usually high frequency components. So this is something we could only capture with the flexible FIR set. And this also needs a slightly higher sampling rate to be captured. I don't think we would need the 0.2 seconds here, but the higher the better, the more sensitive you are to effects like this. So here, we don't see this fast decrease, but we can nicely see that the FIR basis set remains flexible over extended period. And so we could really model this prolonged undershoot period here in the left primary somatosensory cortex. In the right primary motor cortex, we found this pattern that resembles an initial dip. It is at the moment not clear whether this is really above uncertainty level, but sort of what I'm showing here are the maximum likelihood estimates, and they show this pattern. And what's really nice about it is that we find roughly 5 samples within this pattern, which is just something we could do due to the high sampling rate. Of course, you don't find this pattern here in the informed basis set. We found the exactly same pattern of this initial dip-like structure in the right primary somatosensory cortex. A stronger variation of the HRF could be found in the cerebellum. So you can see here this primary response of the HRF is clearly widened. And that is actually something the informed basis set and also the FIR basis set can nicely account for. The same could be reproduced in the right cerebellum. So with this, I hope I could convince you that 3D T-Hex spirals provide a powerful approach for rapid fMRI. We could do high whole brain imaging at a really fast sampling rate, and this gave us access to many different brain regions within one experiment. And most results we've seen were consistent with what we know from literature. The benefits of rapid fMRI in general are clearly the high time series fidelity, especially that you can truthfully recover high frequency components. And it also enables the use of extremely versatile models like the 160-bin FIR we could use here. Nice thing about those versatile models is really that they can capture high frequency components like peak narrowing or an initial dip, but also remain very flexible across the whole duration during which you want to estimate the HRF, so we could also always model those prolonged undershoot periods. With that, I think I could show you some interesting things, and of course I would like to thank all my collaborators who worked with me on this project.